today starting with the NEB cutter or NEB cutter, that is mainly the tool which we use to find out the restriction site within our sequence. So, for that we need a sequence. In order to find out a sequence, we should go to NCBI. If you have a gene with your own, if you have your own gene, you can take that gene sequence and you can do with me, so that you can find out what all restriction sites are present in the that gene. Usually restriction enzymes are the enzymes which cut at a specific specific sequences. For example, one of the restriction enzyme is EcoR1. EcoR1 cut at a sequence called GAATTC. That's specific sequence, and a specific mode of cutting is also there for restriction enzyme. So, I don't have much time in giving more introduction about restriction enzyme. So, restriction enzymes are the enzymes which cut at a specific sequence. So, each restriction enzyme cut at specific sequences. So, depending on that, there are different types of restriction enzymes. All these restriction enzymes cut at specific DNA, specific DNA sequence. So, in order to find out the restriction enzyme cutting site present in a sequence, that is what NEB cutter tool is about. NEB cutter find out how many restriction enzyme cutting site in your sequence which you are uploading in that tool. Actually, the NEB cutter is a bit more commercial thing also, because NEB cutter looks for the restriction site throughout your gene, as well as it will display the restriction enzyme available to that company. NEB means New England Biolabs. That company has been released or developing restriction enzymes. So, these restriction enzymes, they will display whichever restriction enzyme they have available in their company, also from the other companies they will display, so that while you are mapping the restriction site present in your sequence, they will tell you what all restriction enzyme we can use to cut your specific sequence. We can take just a gene, okay. You will understand as we will cut. We will take a nucleotide, we will go to GenBank, and I will take trypsin. Any gene you can take, common genes which come to my mind only I am just taking. So, when I am taking trypsin, you can go to get a trypsin gene here, mRNA. So, I am just taking the FASTA sequence. For that, this is the gene sequence of trypsin from this Manduka sexta alkaline midgut trypsin. So, this we need to know what all different types of restriction sites present in this sequence, so that we can use that restriction enzyme to cut this sequence, so that we can use that restriction enzyme to cut this sequence, so that we can clone this particular gene in a plasmid, or else we can use that restriction enzymes to cut the sequence, so that we can use this as to study the restriction analysis of the particular gene. So, for all this purpose we can use this if we found out what all restriction sites are present inside this gene, okay. So, we are going to find out using a tool called NEB cutter. NEB cutter is a tool which is maintained by New England Biolabs, that is called NEB cutter. So, their latest tool is this, and this New England Biolab company is releasing, or they are the manufacturers of these restriction enzymes. So, they will, if we enter our sequence here, okay, we can enter our sequence, paste our sequence here. if we enter our sequence here, they will find out how many restriction sites are present within our gene and which restriction enzyme can be used to cut our gene. We can enter like this by copy pasting or typing here, or we can enter like this by browsing the file. We can upload the file using we can save the or paste this sequence in a a notepad, and then we can upload it here, or we can just give the GenBank accession number, so they will directly fetch this. You can give this accession number so that they can directly fetch this sequence from the GenBank. Or next is the vector. So they have a plasmid vector and a viral phage vector sequence in their databases NEBCUTTER has. So if we want to know, these three are about uploading our sequence and finding out the restriction site present in our sequence. Okay. In case of plasmid vector and viral and phage vector, we are just finding out which the restriction sites present in a model plasmid vector or viral phage vector. If you just click this drop down, you can see different types of plasmid vectors which is commonly used: litmus, PACP, GPI. So a lot of plasmid vector commonly used. You can see the common vector PBR322. So commonly used plasmid vectors we can see here, all commonly used plasmid vectors. If you click that, the restriction sites present in that plasmid vector we can see. You can just click PBR322, which we all know. Here is PBR322. PBR322, which you people all know this. We will come to the sequence later. So in order to know the restriction site present in the plasmid, we can this PBR322, we need to set the preferences. We can set the preferences whether our genome is linear or circular, etc. Or genome, this PBR322 is circular only. So we can click circular, and you can name the project. It's optional only. You can click here. I'll come to our sequence. Yeah, we are just seeing the PBR322. You can see, I think this picture you have already seen in your textbooks all. What all types of restriction enzyme can cut the PBR322. All the restriction site present in the PBR322 will be displayed. So if you are cloning your gene using PBR322, you can select any of this restriction enzyme. You can they are saying, they are denoting color codes which indicates that some of them are blunt and cutting enzymes, some of them are sticky and cutting enzymes. Usually restriction enzymes cut two types of cutting they will make. One is blunt and cutting, and second is sticky end cutting. So blunt end cutting means sorry, I will show you, because taking full restriction enzyme is not possible in this class. So DNA sequence, if they cut by restriction enzyme, some of the restriction enzyme makes blunt end cuts, means as they cut we will get two blunt ends. This sequence, when they cut here, we will get two blunt ends. So you can see like this. Some enzymes cut sticky lead to sticky ends. Some enzymes cut lead to sticky ends. You can see some enzyme cut like this to get sticky ends. So some enzymes, sorry, cut to get sticky ends, the ends which can easily get ligated, sticky ends. So some enzyme give sticky ends, while some enzymes give blunt ends. So these type of two types of cut these restrictions in can bring about. So which enzyme cut in which way they will tell us this NEB cutter. Blunt and cuts, how they cut, cuts one strand. Some of the enzymes cut only one strand. This sticky and 5' extension, sticky and cuts with 5' extension, sticky and cuts with 3' extension, and they will give particular color so that that enzyme is present in NEB, so that you can buy from NEB. Some enzymes may not be present with the NEB, so you can buy from another suppliers, and some enzymes may not be commercially available. So linear, circular, all these things they will be displaying. You can zoom it and zoom out and see these restriction sites. So this is all about one of the uses of the NEB cutter or NEB cutter. Actually the company uses the name NEB cutter, but I have heard many people using it as NEB cutter. That is why I so the company name is such NEB cutter only. But many people working in this genetic engineering, they use this as NEB cutter. So if you need to select some viral or phage vector, you can see examples of viral and phage vector, lambda phage, all vectors are available. You can just check with the viral and phage vector here, and you can find out the restriction map of that vectors here. So that is not our case. We are just adding a sequence so that in order to find out restriction site present in that sequence. That is our main aim. So these are the these are the things which you can do in a NEB cutter. So our genome is not our gene is not circular, it is linear only. We are not clicking the circular, and we are just submitting it so that you can see our genes, our gene which we have given, this the trypsin gene which we have given consists of how many nucleotide? 804, is it? 804 bases, 804 bases which contain these many restriction sites. So these enzymes can be used for cutting this gene according to your use. What is your use? You can use this restriction enzyme. You can select this any of these restriction enzyme to cut your gene sequence. So this is how it get displayed. If you the same thing, all these blunt and cuts, sticky and cuts, all are displayed in this color. You can just zoom in so that you can see it more clearly. Yes, you can zoom in so that you can get a more clear idea. Each codon is represented by the amino acid name, which codes by each codon is being represented here yellow. You can see how they cut. You can this MSP J1 enzyme, how they cut. You can see here one cut and here one cut. It is a sticky end cutting it is showing. So here one cut and here one cut. So it will give that fragments. So like that we can give we can get a close look on the whole genome sequence. You can just scroll it so that you will get a close look on the each nucleotide sequence and how the enzyme cut, the very detail about how the enzyme cut. All these things were displayed in this and if you need to buy from NEB or any other supplier, you can buy and give order here also. So it is a and you can see this tool can help in NEB getting more orders also. So you can see it also indicates the, you can see here star, the symbol here you can see star and hash and this bracket ambiguous site. So you can see whether this restriction site can may be methylated. Any epigenetic modification can be methylated, CpG methylation or other type of methylation can occur in this restriction site. That also it will be indicating. So you can see star hash symbol, which indicates you can see some enzymes, the enzyme recognition site could be methylated, so that it can affect the restriction, the cutting of the enzyme. So like this, all details about the restriction enzyme cutting site or a particular gene can be mapped using a NEB cutter tool. Next, one of the other things you can do is you can go to the custom digest. Here the NEB in the cutter you can have a link called custom digest. You can digest your gene which you have given, this gene. You can digest your gene in silico. You can digest your gene using a restriction enzyme and see that see the bands after the restriction digest in a agarose gel. All can be done with the in silico. You can select one of the enzyme or two of the enzyme depending on your use. So I will select one enzyme only. BAM-H1 I can, BAM-H1 I think this is two enzymes included one. So this BAM-H1 I am selecting. So this BAM-H1 I am selecting, and these are how many cuts it can make, all details about the enzymes. All we have seen all restriction enzymes which can cut that gene sequence. That all restriction enzymes has been shown in this table, and their recognition site, as I said, people who have not studied restriction enzymes cut at a specific sequence. These cut only in this specific sequence. So like that, how many cuts it can make, so like that, its activity, all these are mentioned in this table. So we are just selecting one of the enzyme, and we are going to cut our gene with our enzyme. Click that enzyme and click the digest, so that our enzyme, our gene will be digested in silico by that enzyme, and we will get restriction digest in a agarose gel. So before going into lab and doing the restriction digestion, we can do it in silico and confirm that how many what if we cut that gene with echo R1 or cut that gene with BAM-H1, how many fragments you are going to get. You will get it first, then you can go for it doing in the lab condition. Just click digest. So it will be digested. It is already digested. It is BAM-H1 cut here only. It gives BAM-H1 cuts at 303. So it will give 2 fragments only. Is it? 2 fragments only. We need to see the 2 fragments. Just click gel here. So you can see the 2 fragments here. BAM-H1 cut at coordinates 304 1 to 303. We can see the length of 303 base pair, and next is 303 to 804, that is 501 base pair. You can see that 501 here and 303 here. So you can see, if this is the agarose gel, this is 0.7% agarose gel. If you are using 1% agarose gel, the band may the formation of band or the band position may change. So you can try whichever percent of agarose gel you are using here and find out where your band will come. Okay. So everything you can do virtually, and if you are not using agarose gel with the TBE buffer, you can get another result, or other type of gel, Spreadex gel, polynat gel, etc. You can check that, so that you will get different pictures yeah, different what the movement or the position of the bands will change with the percentage, all these things. You can see the band, the position may slightly changes. And if you need to add a ladder, you can add a ladder also. You use usually 1 kb ladder. 1 kb ladder you can add, so that you can see where this will come, these bands will come. So all these things can be done in silico condition, and you can analyze your bands, and all these things can be done using this NEBCUTTER. It's a very useful tool if you are working in genetic engineering, or if you are working in molecular biology itself. It's very useful tool. It can be utilized very effectively. So this is all about NEBCUTTER. Hope you understood. Hope you all, yes, today, yes, many people have joined later. So now we will go into sequencing. This is about NEBCUTTER, and we have found out how it cuts, and how it can be shown in gel. So this is how it will be done. So these are some of the tools which we can use it for research purposes. I think